Bismillahirrahmanirrahim. Today the topic of discussion is neoplastic proliferation of white cells. What does it mean that there's a malignant conditions of the white blood cells, and as a result there is an increased proliferation of white cells and their count is increasing. There are three types of the neoplastic proliferation of white cells and neoplasms of white cells. What are those three? Either there's lymphoid neoplasms. What are those called? Lymphoid neoplasms. The second is myeloid neoplasms, and the third is histiocytosis. Yes, histiocytosis. Okay. Now, in the lymphoid neoplasms, there are further three categories. If they are originating from the B cell, then it would be called B cell lymphoid neoplasm. If the T cells are involved, then it would be called T cell originating. And the third variety is the natural killer cells. Okay. And in the myeloid neoplasms, there are also three categories. The first is acute myeloid leukemia. Acute myeloid leukemia, AML. The second is myelodysplastic syndrome. Myelodysplastic syndrome. And the third is myeloproliferative disorder. Chronic myeloproliferative disorder. And histiocytosis, it is rare. It is mainly the proliferation of macrophages and dendritic cells. It is a proliferative lesion of mainly the macrophages when macrophages are multiplied, and the second is the dendritic cells. Now in acute myeloid leukemia, there's basically the immature progenitor cells accumulate in the bone marrow. In acute the detail would be discussed in the later lectures. Mainly in the acute myeloid leukemia, there are immature progenitor cells that accumulate in the bone marrow. While in myelodysplastic syndrome, dysplastic means ineffective, insufficient. So in myelodysplastic syndrome, there is mainly an ineffective hematopoiesis. Ineffective hematopoiesis or synthesis of blood cells. So as a result, there would be peripheral blood cytopenia. What would occur? Peripheral blood cytopenia. Whereas in chronic myeloproliferative disorder, proliferative means an increase in the count. So in myeloproliferative disorder, there would be increased production of one or more the terminally differentiated myeloid elements. Okay, there would be an increased production. So what does it occur? There would be an elevated peripheral blood count. Elevated peripheral blood count. Okay, these were the types of the neoplastic proliferation of white cells. Next we discuss about the etiologic factors of these WBC neoplasms. What are the etiologic factors? That what are the underlying causes or the mechanisms of these neoplasms? There are mainly 6 factors. We will discuss one by one. The first is chromosomal translocation. The first is chromosomal translocation or other acquired mutations. These are mainly the acquired mutations. Okay. As a result of chromosomal translocation, the genes get mutated. Now these mutated genes, either there would be a gain of function mutation or a loss of function mutation. Either there would be gain of function mutation or loss of function mutation. In gain of function mutation, there is an inappropriate increased function of the normal gene. whereas in loss of function mutation there is decreased function of the normal gene. So resultantly the aberrant proliferation of these white cells occur, so the neoplasms occur. The second is that as a result of these mutations, the oncogenes are get activated, and what they do, these oncogenes form the oncoproteins, and the three main mechanisms of oncoprotein as a result the neoplasms occur. What are those? These are either they arrest differentiation of the cells at a stage where the cells are dividing rapidly. What they do, they arrest the differentiation of cells at a stage where they are dividing in a very rapidly, in a very fast speed, so the number of those cells would increase. And the second mechanism is enhance the self renewal. Whenever the precursor cell is differentiated into a next stage, then it also form a daughter cell which regenerate to form the parent cell, so that the number of precursor cell does not get depleted, and these oncoproteins they enhance that process, that self renewal. And the third mechanism is inhibition of apoptosis. Inhibition of apoptosis. When the apoptosis would inhibit, again the number of the cells would markedly increase. These are the three main mechanisms. Now the second cause is inherited genetic factors. Second is inherited genetic factors. These inherited genetic factors, what they do, they basically impart the genomic instability. Okay, these inherited genetic factors they actually impart the genomic instability, so there is an increased risk of developing the leukemias and lymphomas. For example, Down syndrome, neurofibromatosis type 1. These children, they are at increased risk of developing the childhood leukemia, because these conditions impart the genomic instability, so there is an increased risk of developing these leukemias. The third cause is the viruses. There are three main lymphotrophic viruses which are associated with these neoplasm. What are those? The first is human T cell, leukemic virus type 1. Human T cell leukemic virus type 1, HTLV1. The second is Epstein-Barr virus, and the third is human herpes virus 8 or Kaposi sarcoma virus. The human T cell leukemic virus 1, it is associated with the adult T cell leukemia. It is associated with adult T cell leukemia, whereas Epstein-Barr virus, it is associated with Burkitt lymphoma or B cell lymphoma or Hodgkin lymphoma, whereas human herpes virus 8 or Kaposi sarcoma virus, it is mainly associated with the B cell lymphoma. Now the fourth cause is chronic inflammation. In the setting of chronic inflammation, various mutations occur. As a result, there is an increased chance of developing lymphomas at that site. Example is H. pylori infection. It is basically a chronic inflammation of the gastric mucosa. As a result, there is an increased risk of developing gastric B cell lymphoma. And the second is gluten sensitive enteropathy. These patients, they are also at increased risk of developing the intestinal T cell lymphoma. because at that side there's a chronic inflammation going on, so there's various mutations and there's an increased risk of developing these kind of lymphomas. And the fourth cause is iatrogenic factors. Iatrogenic factors mean that for the treatment of other malignancies, various radiations or chemotherapies are given. Radiations and chemotherapies are given, and these radiations and chemotherapy, they cause the mutation of the normal cells, like hematolymphoid progenitor cells, which are the precursor of these various blood cells. And so there's an increased risk of developing myeloid and lymphoid neoplasms. The sixth cause is smoking. In basically tobacco smoke, a carcinogen is present, which is known as benzene, and it causes the various malignancies. The one of those is acute myeloid leukemia. The smokers, they are basically 1.5 to 2-fold at increased risk of developing the acute myeloid leukemia. Now this was all about today's lecture. A brief review that neoplastic proliferation of white cells, what are the main types: lymphoid, myeloid, and histiocytosis. Okay, and lymphoid neoplasms, either it is B cell, T cell, and natural killer cell, and the myeloid neoplasms, acute myeloid leukemia, myelodysplastic syndrome, or chronic myeloproliferative disorder. And histiocytosis, it is a rare but proliferation of macrophages and dendritic cells. In the etiologic factors, there are 6 main causes. Either there is an acquired mutations like chromosomal translocations, the genes get mutated, and there's either gain of function mutation and loss of function mutation, and the oncoproteins by their own mechanism, by arresting differentiation or by enhancing the self-renewal or by inhibiting the apoptosis, and increase the risk of developing leukemias. The second cause was inherited genetic factors. Due to these, there's a genomic instability. For example, Down syndrome and neurofibromatosis type 1 children, they are at increased risk of developing childhood leukemia. Then third was viruses. 3 main lymphotrophic viruses like human T-cell leukemia virus type 1, Epstein-Barr virus, human herpes virus 8, and they are responsible for these respective neoplasms. Fourth cause is chronic inflammation like H. pylori infection, which increases the risk of chronic B-cell lymphoma, gluten-sensitive enteropathy, which increases the risk of intestinal T-cell lymphoma. Iatrogenic factors, the fifth cause is radiation and chemotherapy, which causes mutation of the progenitor cells and they increase form the neoplasms. And the sixth cause is smoking, which contain the benzene and increased risk of acute myeloid leukemia. Thanks for joining.